ABC Digital Healthcare channel. This is a channel where we will be learning basic things in healthcare management. Let's get started. ABO Blood Group and Resource Antigen. We are going to discuss today. ABO Blood Group System. It is the classification of human blood based on the inherited properties of red blood cells, which is the erythrocyte, determined by the presence or absence of the antigens A and B, which are carried on the surface of the red blood cell. Persons may have type A, type B, type O, type AB blood cells. The blood group antigens are actually sugars attached to the RBC. Antigens are built on the red cells. Individuals inherit a gene which codes for specific sugars to be added to the red cells. The type of sugar added determines the blood group of the red cell.
This is the diagrammatic representation of how A and B antigens are being formed of the red blood cell. The H gene, which serves as the substrate in this diagram, is being found on the red blood cell. The N-acetylgalactosamine, when being added to H gene on the red blood cell, there will be production of A antigen. And when the sugar D-galactose is being added to the H gene of the substrate, there will be formation of B antigen. When these two sugars are being added, the N-acetylgalactosamine and D-galactose are being added to the H gene substrate on the red blood cell, there will be production of AB antigen.
Blood group antigens are codominant. If the gene is inherited, it will be expressed. Two genes inherited, one from each parent in ABO blood group system. Individual who is A or B may be homozygous or heterozygous for the antigen. Heterozygous, that's AO or BO. Homozygous, that's AA or BB. Phenotype is the actual expression of the genotype, e.g., group A. Genotype are the actual inherited genes, which can only be determined by the family studies, that is, for example, AO. Example of determining genotype. Mom's phenotype is group A. It has a genotype of AO. And Dad's phenotype is group B, has genotype BO. On this table, if we cross the two genotype, they are going to give us a particular blood group. When you cross A and B, it gives us AB group. If we cross A and O, it will give us group A. If we cross B and O, it will give us group B, and BO and O give us group O. Other examples are AA and BB. When you cross them, you have an offspring 100% AB. BO OO will give us 50% each of B or O. OO and OO will give us 100% O, and OO AO will give us 50% each of A or O. Group O. In group O individual, no A or B antigens are present on their red blood cells. These individuals form potent anti-A and anti-B antibodies which circulate in the blood plasma. You can see it in the diagram. On blood group A individual, no B antigens present. These individuals form potent anti-B antibodies which circulate in the blood plasma. Group B individual, no A antigens present on their red blood cell. These individuals form potent anti-A antibody which circulate in the blood plasma. Group AB. In group AB individual, their red cell has A and B antigens. These individuals possess no ABO antibodies. No ABO antibodies, that is, no anti-A or anti-B. The summary of what we are discussing: blood group A individual has antigen A on their red blood cell and anti-B in their plasma. We'll be able to donate blood for A or O blood group individual. Likewise, blood group B individual, it has B antigen on their red blood cell and anti-A antibody in their plasma. We'll be able to give a blood to B or O blood group individual. Blood group AB individual has antigen A and B on their red blood cell, but no anti-B or anti-A in their plasma. We'll be able to give blood to AB, A, B, or O blood group individual, respectively. Blood group O has no antigen on their red blood cell, but has anti-A or and anti-B, anti-A and anti-B in their plasma. We'll be able to give blood to O blood group individual. Antibody clinical significance. ABO antibodies are immune and will result in destroying incompatible cells, which may result in the death of the recipient. That's hemolysis. Individual transfused with an incompatible blood group will lead to destruction of red blood cell. This may result in the death of the recipient. You can see from the diagram the surface of the red blood cell has surface antigens, and when it's being mixed with the opposing antibody, there's what you call agglutination. and which eventually leads to hemolysis. Now let's talk about the resource D antigens. The resource D antigens is of next importance in resource typing. Resource, that's RH, refers to the presence or absence of the D antigen on the red blood cell. Production of antibody to D requires exposure to the antigen. The D antigen is very immunogenic. That is, individual exposed to it will likely make an antibody to it. For this reason, all individuals are typed for D. If negative, must receive resource D negative blood. The most important population to consider is females of childbearing age. If immunized to resource D antigen, the antibody can cross the placenta and destroy resource D positive fetal cells, resulting in death of the fetus. This is why resource negative women are given ROGAM after birth of resource positive baby. We are going to discuss about the hemolytic disease of the newborn, how it occurs. In the diagram A, the child is resource positive and the mother is resource negative. In diagram B, during pregnancy, fetal resource positive RBC escape into maternal circulation. And in C, the mother produces antibodies to resource D antigens. And in D, for the second pregnancy with resource positive child, results in destruction of fetal D positive RBC.